Hey guys, we are doing here section 12.3, which deals with what we call actually non-Mendelian inheritance, which means these are going to be inheritance patterns that don't follow Mendel's rules. So these ideas that we've been talking about so far in chapter 12 regarding what it means to have a dominant allele versus a recessive allele, when we have homozygous versus heterozygous, some of those will not apply here any longer in this section. So in looking at 12.3, this is part of the reason why I'm kind of separating it out from the quiz. You'll test and quiz on this section separately, but the test itself will kind of involve everything together. So we are covering this as part of chapter 12, but just be aware that it's slightly different from what you've been learning up until this point, okay? So other patterns of inheritance, make sure you got the title there. Learning objectives. This is actually a really short section, but I am going to add in some Punnett squares here towards the end. And so I do want you to spend some time practicing those, and you'll have some assignments that tie in with that also. Okay, but the vocab is not nearly as much as what we've seen in some other sections. So we're going to look at, again, exceptions to Mendel's principles, things that do not fit his rules, and then explain how the environment can affect how traits are actually expressed. So based on what you inherit from your parents, you know, what that may look like and how that is different based on where you're raised. All right. So there are going to be, I think 3 or 4 different type of non-Mendelian inheritance patterns, 4 from what I can see. And these are things that, again, they don't strictly follow Mendel's rules. So unlike what we saw with the pea plants, where we had green peas and yellow peas, or round and wrinkled, here it's not just one or the other. You tend to see sometimes 3rd or 4th or 5th phenotypes kind of showing up. So this is our first scenario here, where we get what looks actually kind of like a mixing or a blending of 2 different phenotypes. So the example that we're looking at here is called incomplete dominance, and it is what it sounds like. Neither allele in this case is what we call completely dominant. And in that scenario, what that means is that when you have an individual who's heterozygous that contains one of each, what would have normally been viewed as a dominant and a recessive allele, well, instead of expressing, just that dominant phenotype like we've been seeing, here what we get in the heterozygous offspring or any individual is going to be a mixing of the two, what would have been dominant and recessive. So really, I don't have a true dominant allele in this scenario. I don't really have a true recessive allele. I get like these intermediates in between them. So I've got here in text, both alleles produce protein products, and those proteins combine to create a third and a unique phenotype. All right, so we're looking at these flowers. These are from plants called 4 o'clock plants, which are kind of our go-to example when we talk about incomplete dominance. But you have a red flowering plant, and when that's crossed with a homozygous white flowering plant, what you see is we get all these heterozygous offspring here that are an intermediate phenotype. So meaning they're not red, they're not white, but they're pink. And so I tend to think of incomplete dominance as kind of this blending, often like you would blend paint colors together or something similar to that, okay? I'm going to come back and set up a Punnett square with you in a little bit dealing with this, all right? And I'm going to do that separately with a view of just the board and not the PowerPoint. But we'll go through the notes first, and then we'll do the Punnett squares, okay? Additional example of this is with betta fish, and it's supposed to be this one, which is kind of this greenish color, and this steel blue color. When those two combine, these are the homozygous, somehow you get this royal blue color, and it doesn't quite look like these two would mix and create that, but that's kind of the setup with that. So again, you have 3 distinct and unique phenotypes, and those 3 phenotypes are created by the interaction of what are not completely dominant alleles, okay? The second scenario here is something called codominance. And you got to be careful with this one because it sounds a little bit like what we just saw on the last slide. What makes this one different is that we do not have a blending of traits. We get both alleles that are equally powerful in a way. So both alleles are considered dominant, and as a result of that, both are fully and separately expressed. So there's not one that covers up the other. They just show up. Right? Like they consistently will show up when you have an individual who's heterozygous. So you can see again, both alleles here produce protein products, but in this case, they're not mixing to create a third and unique phenotype. Here, those products remain separate, okay? So you can see how this differs from what we saw in the example with the flowers on the last slide. We're not blending. If we were blending black and white, we would get a gray chicken. Here we're blending black and white, and we get what's called a checkered chicken because they have both black and white feathers, and they have some unique coloration patterns, okay? We see this in humans more frequently in things like blood types, all right? And this will be one of those Punnett squares I'm going to help you with here in a little while, but you can see what's happening in this scenario. Humans have four, really at minimum, we could say we have four different blood types. In addition to that, you can be positive or negative, and so that adds some extra complexity there. But if we just think about the letters that we use to represent blood types, you can be blood type A, blood type B, blood type AB, or blood type O. And where that's coming from is based on kind of the mixing and the expression of alleles on a specific gene. And so you can see we have this A allele, we have a B allele, and those produce the A blood type and the B blood type. If you get A and B together, this is where we see codominance happening. We get both the A, what we call antigen or protein being expressed, and the B protein being expressed. And because of that, the person said to have AB type blood because they have this A and this B component. By the way, these are red blood cells, okay? And these antigens are expressed on the outside of those. So if you're type AB blood, this is normal for you. This is what your red blood cells would look like. And if you're blood type O, it's kind of the exact opposite. This is a scenario where we have actually still two recessive alleles. So we've got these codominant alleles, A and B, and then we've got these two that are recessive in comparison to that, so that we still have this opportunity to have blood cells without any antigens or proteins on them. And as a result of that, red blood cells look pretty plain and barren. And, you know, blood types play a big, big role in our immune system in helping our bodies to identify what should be there and what shouldn't be there. So for a person with blood type O that's lacking these antigens, they can run into trouble sometimes if they were kind of exposed to blood type A, B, or AB, if you know they needed a transfusion or something in a hospital. So again, these proteins are produced by the interaction of these different alleles over here. And you've got A and B again that are codominant. They both get expressed fully and separately. And then these little I alleles represent what would create the O phenotype, because there is no A and there is no B in that case, okay? So again, we'll come back and do Punnett squares on that in a second. Another type of non-Mendelian inheritance pattern, and this is the third one, is regarding something called multiple alleles, and this is, again, also what it sounds like. So if we have a scenario where we have more than just dominant and recessive, we can have kind of a range of dominance in the alleles that we find for specific genes. And we see this oftentimes in things like skin color or fur color, like what you're seeing here in this example with rabbits. So we have several different alleles here. And you can see at the bottom, I have several examples. And by the way, you do not have to write out this example, all right? If you find it helpful, then certainly, you know, feel free to write it out, but I'm not going to ask you to write out all of this. So here are my four alleles in this case, and you can see what the color actually looks like, and it also describes, you know, what it's dominant to if there is any level of dominance for that one allele. The most recessive trait is this albino rabbit, and in this case, to be albino and be recessive, you have to be homozygous recessive. For all of these other ones, whatever's most dominant is what we're going to see. So in the case of a rabbit that has one big C allele, the full color allele, as long as they have one copy of that, they're going to be this darker brownish color, okay? Now, if they lack that big C allele, but they have this CCH allele, the one that causes a chinchilla phenotype, as long as they've got one of those, they're going to look chinchilla and gray. The last one, or I should say the third one on the list, but the last one to talk about, C with the H allele. Again, you only see that phenotype if you have a scenario where the rabbit has at least one CH allele and nothing else that's more dominant than that. Okay, so it can be CH and a C and still be Himalayan, right? So in this case, we have not sharing of anything really. We're not talking about, you know, expressing things separately or blending them together. What we're seeing is a scenario where we have more than just two alleles for something, right? And so what you end up seeing is based on which ones are present and how dominant those alleles actually are. Okay. The last non-Mendelian inheritance pattern, not our last slide, but the last one here of this kind of category is polygenic traits. And these are even more complicated than multiple allelic traits. These are, if you remember the term poly, it means many. So we're talking about traits that have many genes, the interaction of more than one gene in order to create these phenotypes. We see this most often in human skin color and eye color and height and weight and eye color. I think I mentioned eye color already, but all of those are examples of polygenic traits because not only do we have, two alleles for each thing, we also have now multiple genes. So this text box here is kind of in the wrong spot. I need to shift that up when I'm done here, but our order of dominance, similar to what we see with multiple alleles, but here again, across different genes, would be brown eyes are where we have the most dominant alleles that creates that as the dominant phenotype. Green eyes are kind of in between, and then blue eyes are the most recessive combination. So we've identified at least 3 genes. They're listed right here. And you can see what the dominant alleles produce in terms of phenotypes. So if someone actually has dominant brown colored eyes, they're going to have the brown allele here, and probably green here. And together, those will create that brown phenotype. If someone has blue eyes, again, all that they can have are these recessive alleles. And so when we think about this, any of those colors, the variations in between, is going to be kind of a mixture of some of these things. All right. And so that's why we don't just have, 2 different eye colors or even 3. There's a lot of variation within that. Okay. So the good thing for you about polygenic traits is that I will never ask you to do a Punnett square about polygenic traits because they are far too complicated. All right. Multiple alleles maybe, but not polygenic traits. Okay. The last slide for this section for 12.3 is about environmental factors and influences. So the last thing we need to just kind of emphasize here as part of our conversation about inheritance is to say that it's not always just what you inherit that creates your actual traits, right? So the environment can also influence certain species and certain traits in different ways. One of the best examples of that is in the picture that you're seeing right here. So sea turtles. The sex of a sea turtle is determined not based on chromosomes. This is kind of a strange thing. They are definitely unique in this way. But sea turtles, what they actually end up doing in order to determine sex and whether or not they'll be male or female is when the eggs are laid and kind of incubated on the beach, the temperature of the beach determines what those turtles all will become. The cooler it is, the more likely they become male. The warmer it is, the more likely they are female. And so then the question is, well, if all these turtles are born at the same time, aren't they all going to be the same gender? Like that would be a problem, right? You got to think about the number of sea turtles and the number of different beaches and the length of the reproductive cycle, right? It's not all at the same time. Temperature is not going to be consistent all the time. So you still get males and females, but in this case, it's highly dependent upon where those eggs are developing and the climate in that area, okay? Human height and weight, we know that is highly dependent upon environmental influences. It is certainly not genetic, right? You can make certain dietary choices or exercise choices, and your height and weight will, correlate to that, right? Coat color in Siamese cats, if you've ever seen these cats, they're the ones that have the black faces and the black paws and the black tails, black ears. The reason for that is they actually express genes differently in those areas of their bodies. So those areas compared to their core, to their bellies and, their backs and the parts of their legs and that are closer to their core areas tend to be warmer. And so those warmer areas remain white or at least lighter in color. The areas that are their extremities, kind of like for us, right? My hands are always cold. My nose is usually cold, even if I don't feel cold. But those are the extremities that we have, right? And so for cats, Siamese cats specifically, what they do, they turn on certain genes to create dark pigment, like the black colors that you see. And if you remember what we talked about with photosynthesis, the darker the color, the more light is actually being absorbed. by that fur. And so it's a way that they have to try and keep warm on those extremities by kind of absorbing more sunlight and keeping that as heat. Okay. Last example, and maybe the best example of this, the one we probably don't think about the most but makes the most sense, is plant growth. So in other words, a plant's not going to grow well if it doesn't receive nutrients, water, and sunlight, right? And so even if a plant has the best genes on the planet, it doesn't matter what those genes are if it doesn't receive those environmental kind of factors and stimuli that it needs to grow correctly, right? If it's growing in really poor soil but has very good genes, it's still not going to grow well. If it has really good genes but it experiences a drought where there's not enough sunlight or not enough water, to be able to do photosynthesis, then it's not going to grow very well either. So plants are highly susceptible to environmental factors, as are most living things. All right. So I'm going to switch gears here and get up and do some Punnett squares back here. So if you want to take a break, and I will be right back. So let's start with the incomplete dominance and the codominance ones first, because those are a little bit simpler than what we see with some of the multiple alleles. So as I'm doing this, it would be really great if you will also jot these down on your notes. All right. So when we think about incomplete dominance, remember what we said is we're going to get a blending of two separate alleles. All right. And I like to really think about it like paint colors blending. That's how I've always thought about it. So the example that was given in the notes was of a type of plant called a 4 o'clock plant. We'll do the same here, but we're going to use snapdragons as our example, which is going to be an example that you see on a homework assignment. All right. And snapdragons, by the way, are types of plants. All right. In case you wondered what that was. So the alleles that we're going to work with are similar to what we saw in the 4 o'clock plants. We're going to use flower color. And in snapdragons, red codes, big R is going to code for a red phenotype. In this case, I'm going to use a different letter, even though I'm technically representing the same type of gene and the same trait, because I want to emphasize that these are not going to follow Mendel's rules, right? So I can't just put big R, little r, because there isn't a true dominant allele and there's not a true recessive allele. So in this case, I'm going to use W to represent a white flower phenotype, all right? So if we look at snapdragons, and let's say that I want to cross two snapdragon parent flowers that have these two genotypes. All right, so we could say these are my parents. So we have RW crossed with RW. And so what that tells me based on this is RW, given the fact that I'm saying this is blending, RW is going to represent a pink flower, okay? RW over here also is going to be pink in terms of its phenotype. And if I set up and, identify my gametes, I know each parent's going to give me one R or one W, okay, because the parents are going to be the same. Based on that, I can set up a Punnett square, kind of real quickly here, representing what the offspring would be from this type of cross. And what I see here is that I get 3 different genotypes, right? We've seen crosses like this before. But again, keep in mind what's happening here is we're not going to follow Mendel's rules with this, right? So we still get a genotype ratio that's going to be 1 to 2 to 1. So I get 1 big R to 2 big RW to 1 WW. That's going to be the same. My phenotype ratio, though, is not going to be the same in this case. Whereas before I would say, okay, these guys both have the dominant allele, right? I'm going to combine them, 75% have that trait. I can't do that here because my phenotypes are different. They don't follow his rules. So I know anything that has big R is going to be red. So I'm going to say 1 red. But now I have these RW phenotypes or genotypes. And like what I set up here, RW is going to represent the pink flower at this point. So I'm going to actually put down 2 pink and then 1 white. Okay, so truly it's just like what we did before. The difference here is just simply that In this case, we are not getting just two phenotypes, right? We're actually blending some things, which was not what he saw with his experiments, okay? So it's incomplete dominance. If you didn't jot it down, go ahead and do that. We're going to do kind of the same scenario, and I'm going to erase this, for codominance and kind of look at how these two compare. So even though snapdragon flowers are not something that is going to exhibit a codominance type of relationship, I'm still going to use it here because I do want to be able to kind of directly compare these two, okay? So codominance was the second. Let me go ahead and put that down up here. All right, and so if you remember the difference between these two, something that's codominant is not going to blend so much as it is going to just fully and separately express the actual traits. So when I have a heterozygous individual here, we don't blend full, we have full and separate allele expression. Okay, and so that means I'm going to get to see actually here now two dominant traits appearing at the same time. So again, I'm going to stick with the same alleles, big R, big W. R again, I'm still going to keep as red. W, I'm going to remain and keep as white. Let's set up the same two parents again so we can kind of directly compare this. So we'll do RW crossed with RW. Okay, same gametes, right? Each parent can give one R or one W. So if I set up my Punnett square based on that, I'll get really the same genotypes here. That's not different, right? Anytime we cross two heterozygous individuals, we get the same general ratio of this stuff, but what's going to differ here is how we interpret that in terms of a phenotype. So in codominance, again, we get full and separate allele expression. This creates things like spots or stripes or something like that. So my genotype ratio here, same as what we saw with incomplete dominance. I'm going to have 1 big R, 2 big RW, 1 WW. Okay, that's still the same. Phenotype ratio, though, is going to differ. So I'm still going to have 1 red. But now instead of having, instead of having 2 pink, I'm going to have 2 red and white spotted. Okay, so they're going to express both the red trait and the white trait at the exact same time. And so the flowers themselves are going to actually look like they're spotted. And the last one over here, WW, is going to represent, again, 1 just regular white flower. Okay, so just like incomplete dominance, the piece that's going to differ, the piece that matters, is this here, my heterozygous group. They're kind of in the middle. Okay, so what that means for you when you get to a quiz or test, pay attention to what I'm asking for, if it's codominance or incomplete dominance, and use that to interpret what you're doing. Otherwise, Punnett squares are the same Punnett squares we've been doing all along. Okay, so switching gears a little bit, let's look at what it means to have multiple alleles. And in that context, I'm also going to look at blood types because that's really maybe the best example we can use for this. Blood types, remember I said in humans we can have several different types. And at minimum, so we'll do, let me go ahead and label this. Multiple alleles, and this is also going to be a great example of codominance. So I'm going to put both of these up here. So when we talk about blood types, like I mentioned in the first part of the screencast, humans can have 4 different blood types at minimum, right? You can be A, B, AB, or O. And then within those categories, you can be positive or negative for each one. So if we're going to go down that road here and say, okay, this is going to be my example, human blood types. If we ignore the part about positive and negative, right? If we're just going to focus on, the A and the B part of things, then what we can identify is 1 gene, but 3 different alleles that create different phenotypes. So the first allele is the A allele, and this is going to create type A blood. Okay, and I should probably put type A. Okay, the second allele is the B allele, and this is going to create type B blood. Now, I did put an I up here, like a capital I, and I put the A and the B as kind of like a little exponent, like a superscript up there. And the reason for that is we want to be able to show that these are codominant to the recessive trait, to the recessive quality. And the recessive quality, then, if I'm going to use I's, I can use a little I here to represent that. And a little I allele codes for type O blood, which essentially means 0. Like you have 0 antigens or proteins being expressed on your red blood cells. Okay, so these two here, codominant, and then this one here is recessive to all other alleles. Okay, so if they're codominant, remember what that means is we're going to get spots and we get full and separate expression, and that's exactly what happens in blood type. Okay, so if we have an individual that is this, they're going to be blood type A. If we have an individual that's this, they're also going to be blood type A, right? Because they have that A allele, and they either have 2 of them and are homozygous, or they have an A allele and a recessive, and they're still going to be A. Similar to that, if I have the same scenario with B, I'm going to have blood type B. I can do something very similar for blood type O in a way, right? I mean, O we know is recessive to everything else. So if you're a blood type O, your only real option is to be little I. That's kind of the simplest one. And then we have one additional option in blood type. Remember I said we have 4, which is that we have some individuals that are type AB. And so that's where the codominance piece comes in. So if you have someone who's AB, what that means is that they have both the A allele and the B allele, and together that creates blood type AB. Okay, so those are really our options and the genotypes that are used to create these different kinds. These Punnett squares are just like every other Punnett square we've done. It's just now we have this additional piece of complexity here in the fact that we have 3 alleles. All right, so that's what we call multiple alleles. So we think about a Punnett square. Let's say that I have parents that are, let's have one parent that's going to be blood type AB, okay? And I'm going to cross that parent with one who is blood type O. All right? So again, to be blood type O, you have to be homozygous recessive. To be blood type AB, we would technically say you're heterozygous for that trait. And if I figure out my gametes, again, this parent can give either the A allele, the B allele, and then this parent can either give the little I or the little I, right? There's no other options there. So if I set up a Punnett square for that, with those alleles kind of separated here, my resulting genotypes and phenotypes, I'm really only going to get 2 different options. All right, so if parents are A, B, and O, there's really only 2 options in terms of what type of offspring they could create. So I have 2 over here. The genotype ratio would represent these 2 and these 2, right? So for every one of this, I have one of these. We could say we have 50% big A, little I, and that's compared to 50% big B, little I. Okay, that would be genotypes. If we think about phenotypes, well, we know that big A, little I up here is going to code for blood type A, and big B, little I is going to code for blood type B. So I could say 50% type A and 50% type B. Okay. So that's kind of how that works. Now, let's do another one where maybe we don't get 50-50 ratios, okay? So let's look at what that might look like. I'm going to stick with blood types, all right? And I'm going to stick with, two parents, but I'm going to give these parents now two different genotypes that I know will give me some differences in the offspring, okay? So in this case, I'm going to cross one parent who is blood type A, but is heterozygous for that A trait. Then I'm going to cross that parent with one who is heterozygous for the B allele. And what I'll get from that when I set up that cross is I should end up with 4 different genotypes, actually. I'll end up with 4 different potential phenotypes as a result of that. And I get a number of different possibilities here in offspring. Okay, so you can see how those all differ now. I have blood type AB, blood type B, blood type A, and blood type O. So if you go to set up that and then write out genotype and phenotype ratios, well, it becomes, 1 to 1 to 1 to 1 for this, right? There's a 25% chance of producing any one of these genotypes and phenotypes. There's nothing we can combine here, right? We're blood type AB, B, A, and O, right? So, I mean, this is like 1AB to 1A to 1B to 1O. Like, that's it. It's 25% chance. So, where we have a lot of variety is coming in from these multiple alleles, but, again, as I said a few minutes ago, the Punnett scores are exactly the same, okay? So, there's nothing that's changing here. It's just a little bit more complicated. It's a little bit different now in terms of how we're interpreting, these genotypes and what they represent. But beyond that, it's the same stuff we've been doing. Okay, so please don't let it overwhelm you. Think about these as examples of different ways that stuff can be inherited and shown in a phenotype. But beyond that, don't get bogged down in this and hung up on it. Okay. If you have any questions, let me know. Otherwise, I know we'll be doing some practice and, go back and rewatch this if you ever need any help. Okay. Bye, guys.